Good morning, student. Today we are going to know about Sphenopsida, that is Equisetum, the plant Equisetum, which is belonging to the class Sphenopsida. We are going to know about its habitat, structure, reproduction, and complete life cycle. So here is the Equisetum sporophytic plant body, which shows sterile shoot, fertile shoot, strobilus means cone, leaves, nodes, internodes, and rhizome with a small roots and tubers. This is the complete transverse section of the aerial branch through internode. And this one is the LS of a strobilus. This one is the transverse section of a strobilus, and this is the basal portion of the strobilus. Likewise, when there is a development of antheridium, the plant body going through such developmental conditions, that is single antheridium, then single spermatozoids, and this is a archegonium, mature archegonium. While if the equisetum is a dioecious, then female and male gametophytes are found, and if it is a monoecious, at that time both archegonium and antherizoids are found on single thallus body. So in this way, we are going to know about a complete life cycle of a equisetum. So first of all, habit and habitat of equisetum. It shows the plant body of equisetum has an aerial part and underground part, that is called as a rhizome. Rhizome is perennial, horizontal, branch and creeping in nature, while aerial part is herbaceous, usually annual. Majority of the species are small with a size range in between 15 to 60 cm in height and 2 cm in diameter. So this is all about a morphology, and this equisetum is classified into a kingdom, that is a plantae, then subkingdom, that is a tracheophyta, means structural elements are developed over here. Then superdivision, that is a pteridophyte division called as Equisetopsida. Equisetophyta class is Sphenopsida, subclass Equisetidae, order Equisetans, family Equisetaceae, and genus is Equisetum, species the Arvensis. So this is all about a habit and classification of a equisetum. While some species are also grown in higher heights, some species are rarely having a relatively thin stem, while some are showing vine-like habit or climb over adjacent forest trees. It generally grows in wet or damp habitats and particularly common along the banks of the streams or irrigation canals. Sometimes the species of equisetum are indicators of mineral content of the soil in which they grow. Some species accumulate gold about 4.5 ounce per ton of dry weight. Thus, they are considered as a gold indicator plant. So, this is the plant which has capacity to absorb gold particles whatever found in a soil. Hence, these plants help in prospections or exploration for new or deposits. In equisetum, Silica is deposited on the outer wall of the epidermal cells, giving the characteristic rough feeling. Thus, it provides a protective covering against predators and pathogens. So, this is the complete sporophytic plant body of a equisetum. This diagram showing two different type of leaves. First of all, sporophytic equisetum plant body is differentiated into stem, root and leaves. So, stem of equisetum has two parts, that is a perennial underground much branch rhizome and an erect, usually annual aerial shoot. Branching is monopodial. Shoots are differentiated into nodes and internodes. In majority of species, all the shoots are alike and chlorophyllous, and some of them bear strobila at their apices, which is as shown in figure. So this is the apices which shows development of a strobilus. Some shoots are profusely branched, green, chlorophyllous and purely vegetative. The others are fertile and branched, brownish in color, chlorophyllous and have terminal strobili. So this is the vegetative plant body which showing a green leaves, while this is a fertile plant body which shows brownish unbranched chlorophyllous shoots with a terminal strobili. Underground rhizome and the aerial axis appear to be articulated or joined due to the presence of distinct nodes and internodes, and that's why externally the internodes have longitudinal ridges and furrows, and internally they are hollow tube-like structure. The ridges of the successive internodes alternate with each other, and the leaves are normally of the same number as the ridges on the stem. So in this way, there is a two different distinct type of shoot system, that is vegetative and fertile. Internal features of the stem. When we are taking a transverse section of the stem, it appears wavy in outline with ridges and furrows. The epidermal cell wall are thick, cuticularized and have a deposition of a silicaceous material that can be shown in the next diagram. So this is the transverse section of a stem or rhizome. So that structure, that transverse section shows stomata which are distributed only in the furrows between the ridges, and the hypodermal sclerenchymatous zone is present below each ridge, which may be extended up to the stele. Stele means vascular bundle. The cortex is differentiated into outer and inner regions, which is shown in the diagram. So this was the stele, this is the pith cavity, and this complete structure is nothing but a cortex which can shows a canal. So the outer cortex is sclerenchymatous. This is the outer cortex which is made up of by the thick walled cells, that is a sclerenchymatous, which can possess a sclerenchymatous cells, while the inner cortex is made up of by the thin walled parenchymatous. So these are the thin walled parenchymatous cells. There is a large air cavity in the inner cortex, which is corresponding to each furrows and alternating with the ridges known as vallecular canal. These are the lysigenous canal, which extending the entire length of the internodes and form a distinct aerating system. New leaves and branches of the equisetum are produced by the apical meristem. However, most of the length of the stems are due to the activity of intercalary meristem, which are located just above the each node. The activity of the intercalary meristem causes rapid elongation of the internodal region. So these all details are shown in this diagram. The stele is ectofolic siphonostele, which is surrounded by an outer endodermal layer. So this pith cavity is nothing but an siphon, and around them there is an arrangement of a stele. That's why it is called as a siphonostele, and this is a single bundle. That's why it is a ectofolic siphonostele. An inner endodermis is also present in some species of a equisetum. So this is the outer epidermis and this is the endodermis. The endodermis is followed by the single layer pericycle. So this below this layer there is a single layer that is called as a pericycle. This is the another type of representation of a equisetum species, which can shows furrows and ridges are the differentiated into a epidermis. These are the sclerenchymatous cells. These are the chlorenchymatous cells. Sclerenchyma are made up of for the mechanical support, and the chlorenchyma is made up of for the photosynthesis. These are the scaly leaves which are overlapped on a nodal and internodal portion. So A is the diagrammatic representation and B is the cellular representation. So these, first of all, this is the outer layer that is called as a cuticle. Below that cuticle here is the presence of a epidermis. This epidermis is interrupted by the stomata, and below that there is a presence of a sclerenchyma, which are thick walled mechanical tissues. These are the chlorenchyma. This is the chlorenchyma, and this also is the chlorenchyma, which can performing the function that is a photosynthesis. Then these are the big cavities called as a vallecular canal. This is the complete patch, which is called as a cortex, and below that there is a presence of a endodermis. So this one is the endodermis. This part is nothing but an endodermis. Below that endodermis there is a presence of a pericycle, and below that pericycle there is a presence of a metaxylem. So these are the metaxylems. These are the phloems. This one are the protoxylem, and these are the carinal canal, and at the center there is a presence of a pith or pith cavity. So in this way it has a internal features of the stem. This equisetum is exhibit a xerophytic features. These are ridges and furrows are found in the stem. Ridges are developed and protected by the sclerenchymatous tissues. Deposition of silica in the epidermal cell, that is again a xerophytic characteristic with a sunken stomata. Sclerenchymatous hypodermis is a xerophytic characteristic, while it reduced and scaly leaves, its reduced scaly leaves is a xerophytic character. photosynthetic tissue which are found in a stem, that are showing the characteristic of a xerophytic plants. The hydrophytic characteristic on the other hand are 11 well-developed aerating system like carinal canal, vallicular canal and central pit cavity which reduce vascular elements. So these are the hydrophytic characteristics. So both type of characteristics are found in a equisetum. When we are taking a transverse section of a root, at that time the primary root is ephemeral, that is the cylinder adventitious roots are arise endogenously at the nodes of the stem. So at the nodal portion of the stem there is a development of a small roots. In TS of the root shows epidermis. So this is the outermost protecting and limiting layer, that is epidermis, which again followed by the cortex, and below that there is the outer endodermis and then the inner endodermis. These are the metaxylem, these are the protoxylem, and all are remaining are the phloem cells. So in this way the root of the equisetum showing a typical characteristic. The leaves of equisetum are small, simple, scaly-like and isophilous in nature. They are attached at each node, united at least for a part of the length and thus form a sheath around the stem. So in this way there is a articulation of a leaf around the stem. The sheath has free and pointed teeth-like tips. The number of leaves per node varies according to the species. The species with narrow stem have few leaves, while those with thick stem have many leaves, up to 40 numbers. The number of leaves at the node correspond to the number of ridges on the internode below. So this can shows the total number of ridges and furrows in the stem. The leaves do not perform any photosynthetic function and their main function is to provide protection to young birds at the node. So in this way, stem of the equisetum can participate in the photosynthesis. Then equisetum can be reproduced vegetatively and by means of spores. So in case of a vegetative reproduction, the equisetum shows subterranean rhizomes of in some species, that is equisetum telmetea, equisetum albens, and some can develop tubers. So rhizomes and tubers are that plant part which can helpful for the vegetative reproduction, which on the separation form a parent plant, germinate into the new sporophytic plant body. The tubers develop due to the irregular growth of some buds at the nodes of the rhizome. Then in case of a reproduction, spores are so developed within a sporangia. The sporangia are borne on the sporangiophore, which are aggregated into a compact structure termed as a strobilus, which are found on a fertile branches and called as a strobilus cone or sporangiophore spike. So this one is the strobilus, which is also called as a Sporangiophilus or a cone. So first of all, this is the strobilus, which has annuals and that possesses sporangiophores, means a stalk-like structure, and within a cavity there is a development of a sporangia or spore sacs. This is the central axis. This is the transverse section of a strobili, which shows a flat central axis, and this is the base of the strobilus. So strobilus is a compact of an axis which shows whorl arrangement of a sporangiophores. Each sporangiophore is a stalked structure bearing a hexagonal peltate disc at its distal end. On the outer surface of a sporangiophore, 5 to 10 elongate, cylindrical, hanging sporangia are borne near the periphery in a ring manner. The flattened tips of the sporangiophores fit closely together, which provide protection to the developing sporangia. The axis bears a ring-like outgrowth, so-called annuals, immediately below the whorls of a sporangiophore, which provide additional protection during early development. So this structure, diagram D, can show a long elongated pendulate sporangiophore. The annual has been interpreted as a rudimentary leaf sheath by some botanists, whereas others considered it to be a sporangiophoric in nature, as occasionally it bears small sporangia. So later and come towards the development of a sporangium. The mode of development of a sporangium is eusporangiate, U means true, as it is not entirely formed for a single initials, but superficially cells adjacent to the original initials may also take part in the development of a sporangia. Sporangia are initiated in single superficial cell around the rim of the young sporangiophore. The periclinally division of a sporangium initials forms an inner and outer wall. At that time, the inner cell, by further divisions in various planes, give rise to sporangious tissue. So, in this way, this is the monoecious plant body of a Thallus equisetum that shows the presence of spores with elaters, and this is the diagram B and C, which can show stages of a germination of a spore. So this is the covering called as a exine, which is the outer wall, and this is the entine. And then D is nothing but an monoecious gametophyte, which shows archegonia, which are developed at the basal portion, and the antheridia at the apical portion of a single thallus, while E and F are nothing but two different thallus structure, two different plant body, which shows presence of archegonia, that is a female gametophyte, while the development of antheridium within a single thallus, that is a male gametophyte. In this way, there is a development of two different type of spores, archegonia and antheridium. The structure of mature sporangium is an elongated sac-like structure attached to the inner side of the peltate disc of a sporangiophore. It's surrounded by the jacket layer, which is composed of two layers of a cell. The inner layer is generally compressed and the cells of outer layer have helical thickenings which are involved in sporangial dehiscence. At maturity, the strobilar axis elongates. As a result, the sporangiophore becomes separated and exposed. Then the sporangium split or opened by a longitudinal line due to the differential hygroscopic response of the cell walls. So in this way, the sporangium becomes split or descent into a two half, and the spores are spherically filled with densely packed chloroplast. Spore wall is laminated and shows four concentric layers. The innermost is the delicate entine, which is followed by the thick entine and the middle cuticular layer and the outermost epispore or perispores. In this way, entine means a endospore, exine means the endospore, while exine is the exospore, and the true walls of the, and in this way the outer wall layers of a cuticular layer and epispores are derived due to the disintegration of the non-functional spore mother cell and the tapetal cells. At maturity, the epispore, that is the outermost layer, split to produce ribbon-like bands or strip with flat spoon-like tips. These bands are free from the spore wall except for a common point of attachment and remain tightly coiled around the spore wall until the sporangium is fully matured. So this is called as the elaters, which is shown in the figure. The elaters are hygroscopic in nature. The spores, hygroscopic means they are always having a affinity to the water or water currents or hydroscopic currents. The spores remain moist at early stages of a development, and thus the elaters are spirally coiled around the spores. These spores are dry out at maturity, means the spores when become dry at that time they can be liberated out, and consequently the elaters become uncoiled. These uncoiled elaters become entangled with each other elaters, and through this action of the elaters help in the dehiscence process and also the dispersal of spores in large groups from the sporangium. Elaters of Equisetum are different from those of the bryophytes. In this way, there is a complete development of Archegonium and a Antheridium. Equisetum is a homosporous pteridophyte. The haploid spores germinate to form gametophyte. The germination takes place immediately if the spores land on the suitable substratum. If the spore do not germinate immediately, their viability decreases significantly. The spore swells up, absorbing by water, and shed their exine. Means that spore mother cell having a outer wall, that is a exine, that having capacity to absorb water, and the first division of the spores result into two unequal cells called as a small and large cell. The smaller cell elongates and forms the first rhizoids, while the larger cell divides irregularly to produce prothallus. The prevailing environmental conditions determine the size and shape of the prothallus. If large number of spores are developed together within a limited space, then the prothalliform are of the thin filamentous type but relatively thick, and cushion-shaped prothalli are formed from sparsely germinating spores. Mature gametophytic plants may range in size, form a few millimeters up to 3 centimeters. They are dorsiventral and consist of a basal, non-chlorophyllous, cushion-like portion from which a number of erect chlorophyllous multicellular lobes develop in upward direction, and the unicellular rhizoids are formed from the basal cell of the cushion. So in this way, there is a gametophytic generation is going on. The prothallus bears sex organs and reproduce by means of sexual method. So in case of a sexual reproduction, Equisetum gametophytic plant body bears Anthraedium, that is a male gametophyte, and Archegonium, that is a female gametophyte, or if they are found on a same thallus, that is monoecious. Although some unisexual dioecious thallus members are also reported, but some are initially unisexual and then become bisexual. These early sex determination appear to be related to the environmental condition, that is temperature, light, humidity, and the supply of nutrition as well. In order to explore the sexuality in a Equisetum, observed that some of the fragments of a male gametophyte remained male throughout the successive subcultures under laboratory conditions. Some other fragments produce archegonia, which subsequently born Anthraedia in increasing numbers. This phenomena supports contention that Equisetum gametophytes are potentially bisexual. However, in 1969, Hank observed that gametophyte of Equisetum bogetans were unisexual, means bearing Anthraedia, and never change into the bisexual type to form the Archegonium. However, the initial male gametophyte never become bisexual. In case of cats, in 1920, observed that 50% spores germinate to produce male gametophyte, while the remaining 50% spores produce female gametophyte. Through they do not lose their male potentiality, that is Anthraedia develop later in fertilization phase. He termed this as an incipient heterospory. Such study of sexuality based on the enzymatic analysis revealed the intragametophytic self-fertilization in case of Equisetum urbans. Equisetum is a homosporous and therefore definite sex determination mechanism is absent, but the sexuality demonstrated by some of the members appear to be related environmental factors, and therefore it is termed as an environmental sex determination for the favorable conditions. At the time when we are going to study about Anthraedium development, at that time it shows 4 successive stages, that is stage A, that is development of an anthraedial initial cell, and then These antheridial initial cells again divided in a transfer plane. At that time it can be developed into a jacket initials and the androgonial cell. This jacket initial again divide longitudinally to develop two jacket initials and 4 to 6 androgonial cells. In this way, in case of a D, diagram D, there is a development of many androgonial cell and few jacket initials, and later on this can be developed into a mature antheridium which can show a single antherizoids. As like this, in case of archegonium, a single cell which can become an archegonial initial cell, that cell divide in a transfer manner and develop into a primary cover cell and central cell. This central cell again divide and redivide and develop into a venter canal cell and neck canal cell, while the primary cover cell develop into a neck canal cell. In this way, there is a development of a neck canal cell, venter canal cell and egg cell. Later on, the egg canal cell becomes enlarged in size and venter canal cell develop into a few transverse division, as like neck canal cell divide into a few to many neck canal cells which can protect throughout the neck canal, venter canal cell and egg cell. So in this way there is a development of a mature archegonium. Later on, water is essential for the fertilization, and thus fertilization going to be start. At the time of the gametophyte must be covered with a thin layer of water in which the motile anthrozoids can swim to the archegonia. The neck canal and venter canal cell of archegonia disintegrate into to form a passage for the entry of anthrozoids. So in this way, anthrozoids can passes through the canal of the archegonia, but only one of them can fuse with a egg cell. Thus the diploid zygote is formed, and generally more than one archegonia are fertilized in a single prothallus. In this way there is a development of an embryo. The new sporophyte is going to be start at the time of when the zygote converted into an embryo. So the embryo is the mother cell of the next sporophytic generation. Most pteridophyte, several sporophyte develop on the same prothallus. The first division of the zygote is a transverse division. This results in the upper and epibasal cell and lower hypobasal cell. The embryo is therefore exoscopic in nature, that is, the apical cell is ducted outward and that is towards the neck of the archegonium in a polarity. No suspensor is formed in a equisetum. The epibasal and hypobasal cells then divide at a right angle to the ogonial wall. As a result, torsade quadrant stage is established, which is shown in a diagram A, that this is the quadrant, means of 4 cells are developed within this. All the 4 cells of the quadrant are of different size and shape. The 4-celled embryo undergoes subsequent division, and the future shoot apex originate from the largest cell, and the leaf initials form the remaining cells of one quadrant of the epibasal hemisphere. In this way, the equisetum can complete their life cycle. One cell of the epibasal quadrant and a portion of the adjacent quadrant of a hypobasal region contribute to the development of a root. The first root develops from one of the epibasal quadrant and the portion of the adjacent hypobasal quadrant. Quadrant means a four divisions. The shoot, in this way the shoot can be grows rapidly, and later on root grows directly downward and penetrate the gametophytic tissue to reach the soil or substratum. A number of such sporophytes may develop from a large mature gametophyte if more than one egg is fertilized. So this is all about the life cycle of a equisetum. Here are the complete diagrammatic representation of a equisetum life cycle. So in this way we got some idea about equisetum life cycle, and it may help you to know to learn about equisetum. Thank you for watching my lecture.